Very good evening students. So here we are going to start our new chapter, that is cell cycle and cell division. So far from cell biology we have completed cell the unit of life, right. So from cell cycle and cell division, what are the concepts we need to cover and what are our expected concepts from this chapter, and where we have to highlight and learn, okay, that we have to focus with. Firstly, you should know the complete cell cycle, in which you should clearly learn both the interphase and M phase beta. Okay, interphase and M phase should be clear to you, and after that under interphase you should focus the three sequential changes which are happening under each substage of interphase, be it G1 phase, S phase and G2 phase. Clear, right? And under M phase, if you see, we have two subphases of M phase, like we will be having under both mitosis and meiosis. We have both karyokinesis and cytokinesis. Okay, in meiosis we will be having two sets of karyokinesis and two sets of cytokinesis, okay, of which karyokinesis 1, prophase 1 is the prolonged process, okay, karyokinesis 1 and cytokinesis 1 followed by karyokinesis 2 and cytokinesis 2. Clear, right, ma? So under karyokinesis you have four substages. What are they? Prophase, metaphase, anaphase and telophase. As such, prophase 1 of karyokinesis is so much important beta. You can keep how much ever star marks you want. Fine. In every alternate year from cell cycle and cell division, there is a definite question on the prophase 1 of meiosis. Okay. And what are the questions that we can expect in NEET? Most importantly, match the following type of questions. How you will get though? In which phase or in which substage or event, what event is happening? Okay, in which substage or under which phase, what event is happening? To match that you may get match the following type of question, and directly from the NCERT liners you will get the questions directly from the NCERT liners. And third is, we can expect numerical questions. Numerical questions from this chapter connecting the ploidy, okay, ploidy of the plant kingdom, ploidy from anatomy, ploidy especially from sexual reproduction of flowering plants. Connecting these three dots, you may get the numerical question asking you about mitosis and meiosis. Okay, so what you have to focus in this chapter beta, the events, sequential events which are happening under each phase, and you have to highlight prophase 1 more, and after that you have to look into the numericals connecting three dots like from plant kingdom, anatomy and sexual reproduction of flowering plants. Ploidies are very important. So that is how you need to go through this chapter. I hope the fundamental insights of this chapter are clear to you, right? Yes. Next. Firstly, cell cycle. How can you define the cell cycle, ma? Any idea about cell cycle? How can you define the cell cycle? What is cell cycle actually? You know, yeah, each cell primarily from building up the cellular constituents or dividing its genetic material or increasing its cell size, okay, that sequential events will happen. Once the cell size will increase, next genome duplication will happen, and then what happens beta? Division of the cell, karyokinesis and cytokinesis, and at the end formation of two daughter cells. This complete sequential events comes under the process of cell cycle. What are those events, ma? First is increase in cell size, take down, increase in cell size. Second is genome duplication, karyokinesis, and cytokinesis. Lastly, formation of two daughter cells. This complete sequential events comes under what beta? Cell cycle. So, cell cycle includes two major phases under it. What are the two major phases of cell cycle? Interphase and M phase. Okay, it includes two major phases, interphase and M phase. Fine beta. Interphase is also called as, very good Vansh, very good, that is a proper definition of cell cycle. Continue with the class beta. So, interphase is also called as resting phase, or biosynthetic phase, or preparatory phase. Fine beta. Okay. Interphase is also called as what? Resting phase, biosynthetic phase or preparatory phase. Why interphase is also called as resting phase? Because there is no actual cell division that is happening in the interphase. Fine, that is why it is also called as resting phase. Why is it called as biosynthetic phase? Because there is synthesis of cellular constituents, cellular organelles, and constituents like RNA, proteins will be synthesized in the interphase. That is why it is also called as biosynthetic phase. It is also called as preparatory phase because in this phase complete preparation for actual cell division will happen. Fine. So, these are the synonyms of interphase beta. As such, under interphase, what are the three substages we are having? G1 phase, yeah, G1 phase, S phase and G2 phase. G1, S and G2 phase are three substages of interphase. Fine. Next, under M phase. Under M phase, what I told you, you will be having karyokinesis and cytokinesis. Fine. Under karyokinesis, you have four substages again. What are they beta? Prophase, metaphase, anaphase and telophase. Fine. Now, the framework of the cell cycle is clear to you. What are the two major phases? Interphase and M phase. Interphase is also called as resting phase, biosynthetic phase, preparatory phase. What are the three substages beta? G1, S and G2, and M phase is having karyokinesis and cytokinesis. Karyokinesis is movement or division of the nuclear genetic material which is done under four substages. So, these are the four substages. Fine. And then cytokinesis. Clear till here right now? So, now we will actually look into the types of cell divisions that are happening. Okay. Before going to the types of cell divisions, before going to the types of cell divisions, once let us have a clear idea on the interphase. Okay. Interphase has three substages we have discussed, right. So, in that, what are the synonyms of G1 phase, S phase and G2 phase we will recollect. Yeah, as G1 phase occurs after mitosis, G1 phase is also called as post-mitotic phase. Listen beta. G1 phase is also called as post-mitotic phase or pre-synthetic phase. Fine. Now, you tell me what can be the synonyms of S phase? Post G1 phase or pre G2 phase. What can be the synonyms for G2 phase? Post synthetic phase and pre mitotic phase. What can be the synonyms for M phase? Post G2 phase or pre G1 phase. Take down quickly. So, G1 phase can be post M phase or pre S phase. S phase is also called as post G1 phase and pre G2 phase. G2 is also called as post S phase and pre mitotic phase. Fine. M phase can also be called as post G2 phase and pre G1 phase. Clear, right? Now you see what are the events that are actually happening in each phase beta. G1 phase, if you see, Good evening Abdul, continue with the class ma. So, under G1 phase is also called as what beta? Post mitotic phase or pre synthetic phase. So the primary event that happens in G1 phase is increase in cell size. Okay, most of the cell organelle duplication happens in G1 phase only. Okay, what are the two major events that are happening, ma? First is increase in cell size. Yes, cells are metabolically active in G1 phase. Okay, they will increase in size, and most of cell organelle duplication happens in G1 phase. Okay, and then cells are metabolically active. Clear, right? Cells are metabolically active. These are the three events that happen in G1 phase. Clear, right, ma? Next. So those cells which are continuously dividing, they will directly enter into the S phase. But there are few cells which rarely divide, like heart cells and nerve cells, they will rarely divide. Instead of going to S phase, they will move to G0 phase. G0 phase, which is also called as quiescent center, a quiescent stage or inactive stage. Okay, so those cells which are moving from G1 to, instead of G1 to S phase, they will move from G1 to G0 phase. So here you have G0 phase. So what are those cells, ma? Like nerve cells and heart cells, which rarely divide. Okay, from G1 phase, they will move to G0 phase. Whenever they are resuming back their cell division, they will not enter from G0 to S. They will again enter from G0 to G1 phase. Okay, take down.
So from G0 phase, it will go to differentiation sometimes.
Yeah, from G0 phase, if they are differentiating, they will not divide at all. Okay, if they want to resume back their division, they will again come back to G1 phase. Okay, so take down here those cells which rarely divide. Yeah, which rarely divide. For example, heart cells and nerve cells. Okay, those cells which rarely divide, like heart cells and nerve cells, what happens, ma? They will enter from G1 phase to G0 phase. In G0 phase, the cells are metabolically active, but they don't divide at all. G0 phase is also called as quiescent stage. Yeah, quiescent stage or inactive stage. Whenever they are resuming back their cell division, they will enter again from G0 phase to G1 phase. This point is clear to you, no? This is inactive stage because they are not dividing, we can't consider cells as dead cells, beta. Those cells which are entering from G1 to G0, we can't count them as dead cells because they are metabolically active. But then they are not conducting any division. They are not entering into the next phase of cell cycle. That is why we are calling this stage as inactive stage.
So it lengthens the G1 phase.
Yes, and those cells which frequently divide, no, their length of G1 phase will be less. Take down that point also. Those cells which frequently divide, cells which frequently divide, this you can expect in your statement based beta. Okay, cells which frequently divide will have short G1 phase. Fine. Next is your S phase, which is very important. Not just most of the organelle duplication, most of the cellular constituents are also synthesized and duplicated in the G1 phase. Clear, right? All the events of G1 phase are clear to you, na beta? Next is S phase. S phase is also called as what? Synthetic phase. There are two major events that are happening in the S phase. First is if you consider this as a cell, and a nucleus over here. Inside the nucleus, DNA duplication happens. Okay, inside the nucleus what happens? DNA duplication happens. So this is prior duplication and this is after duplication. I have represented symbolically. Here how many chromosomes are there? 2 chromosomes. Here also 2 chromosomes. So there is no increase in chromosomal number, but DNA gets duplicated. You can consider, you can count it as from 2C to 4C. The DNA content gets increased. In actual sense, you will see chromosome shape clearly in the metaphase. Okay, you cannot see chromosome shape or structure in the S phase or interphase. But for your understanding, to convey that one point key, only DNA is getting duplicated. In the NCERT, they have mentioned a statement that only DNA duplication happens, there is no chromosomal duplication. Okay, so I have represented symbolically like this, and this happens in the nucleus. In the nucleus, this is the change that is happening. Fine, in nucleus what happens ma? DNA content is getting duplicated. In the cytoplasm what happens is centrosome duplication happens. Centrosome and centriole duplication. You have already a clear idea, you already have a clear idea on what is centrosome, right? So from Cell the Unit of Life we have learned centrosomes are non-membrane bound organelles where centrioles will help in the formation of spindle fibers or spindle apparatus, wherein 2 centrioles which are cylindrical shaped and perpendicularly arranged inside the centrosome, they will get duplicated in which phase? S phase. So 2 major events that happen in the S phase beta. One event is inside the nucleus, DNA is getting doubled or DNA duplication happens. In the cytoplasm what is happening? Centrosome duplication happens inside the cytoplasm. Okay. So inside the cytoplasm, centrosome duplication happens. Centrosome duplication or centriole duplication also you can consider. Centriole duplication happens in cytoplasm. Fine. DNA doubles inside the nucleus. Fine. So these are the events of S phase. In G2 phase, whichever proteins are required for the duplication still, okay, for further process of karyokinesis, whichever cellular constituents are required, they will get doubled and synthesized in G2 phase, and the major event is self-duplicating organelles or semi-autonomous organelles will be duplicated or doubled in G2 phase. What are those self-duplicating organelles beta? Yeah, chloroplast and yes, good. Chloroplast and mitochondria. These are self-duplicating or semi-autonomous organelles. Self-duplicating organelles will get doubled in which phase of interphase ma? Self-duplicating organelles will get doubled in which stage of interphase? G2 phase. Okay. Self-duplicating or semi-autonomous cell organelles. Fine. Along with the self-duplicating or semi-autonomous organelles, Golgi bodies also get duplicated. Golgi bodies will also get duplicated during the G2 phase. Fine. And there is one more point to add over here, that histone proteins whichever are required for DNA packaging, right? Those histone proteins will be synthesized in the S phase of the interphase. Histone proteins. Yeah. No. They are synthesized during S phase of the interphase. Good, Raghav, we are very attentive today, expecting the same each day. Yes? Any doubts, Samir? Till here it is clear, na beta? All the events of interphase. Once shall we recollect the events? Yeah. G1 phase, what happens ma? Increase in cell size. Most of the cell organelle duplication happens and cells are metabolically active. For frequently dividing cells, G1 stage will be smaller time, and for those cells which rarely divide, for example heart cells and nerve cell, from G1 stage they will move to quiescent stage or G0 stage or inactive stage. From G0 they will move again back to the G1 phase. Next is S phase or synthetic phase. In synthetic phase, two major events happen. One, inside the nucleus, DNA gets doubled or duplicated or replicated, one and the same beta. They will get duplicated in the nucleus and centrosome gets duplicated in the cytoplasm. Histone protein synthesis also happens in S phase. Next, G2 phase, self-duplicating or semi-autonomous organelles will be duplicated, okay, and Golgi bodies also get duplicated here. You can remember symbol mnemonic in G2 phase is CMG, gets duplicated in the G2 phase, okay. Next, clear? Shall we move on to the mitosis? Before going to mitosis, you should know there are three types of cell divisions, beta. Those three types of cell divisions include, first is amitosis, take down parallelly, mitosis and meiosis. Amitosis is that cell division which mostly happens in unicellular organisms, be it prokaryotic unicellular or eukaryotic unicellular organisms, which is most notedly known as what? Binary fission. Binary fission. What does amitosis mean? That division of two cells without any involvement of spindle apparatus formation or without any involvement of spindle fibers. If two cells divide, okay, karyokinesis and cytokinesis happens just through the division without the involvement of spindle fibers, then it is called as amitosis. So, there is no involvement of spindle fibers over here. No. Fragmentation, regeneration, that is different, ma. So, this mostly happens in unicellular organisms. Best example you can take is amoeba. Okay. Next is mitosis. Mitosis we are going to exercise now. Mitosis occurs in all the somatic cells. Okay. Mitosis occurs in all the somatic cells, and mitosis is also called as equational cell division. This is also called as equational cell division because the number of chromosomes present in the parent will be equal number of chromosomes in the daughter. Both the daughter cells and the parent cells will have equal number of chromosomes. Equal number of chromosomes. For example, parent cell is having 46 chromosomes, daughter cell also will have 46 chromosomes at the end of the division. Whereas meiosis is called as reductional cell division. Reductional cell division. Fine. And this occurs in where? Germ cells. This is called as reductional cell division because chromosomal number gets half. For example, in parent cell, if it has 46 number of chromosomes, daughter cell will have only 23 chromosomes. Okay, half the number of chromosomes, it will be reduced. So, that is why it is called as what? Reductional division. Okay, and one point to be highlighted over here, beta, mitosis occurs in animal cells. Mitosis occurs in the diploid cells only, means those cells which have complete set of chromosomes, only in those cells mitosis will happen. Of course, there are few exceptions, like in honeybee we see. Okay, in genetics when we are looking sex determination, haplodiplontic type of sex determination you will observe in honeybees, okay, wherein mitosis can happen in haploid cells also. Okay, that can happen in animal cells. And in plant cells, both haploid and diploid cells mitosis will happen. So, take down, yeah, mitosis occurrence. In both animal cells and plant cells. In animal cells, if you see, occurs only in the diploid cells. Exception is what? Honeybees. In honeybees, male honeybees, the haploid set of chromosome will have how many chromosomes beta? 16 chromosomes. Male honeybees will have 16 chromosomes haploid set. This will undergo mitosis and they will give again 16 set, half the set of chromosomes. Okay. So, as such males will produce males like this through haplontic division, sorry, through mitotic division. Fine. Next is what? An plant cells. Mitosis occurs both in haploid cells and in diploid cells. Both in haploid cells include what? Gametophytic cells, and in diploid cells also. Yeah, because it shows alternation of generation, right? Both haplontic phase and diplontic phase is observed in a plant life. So, in both haploid and diploid cells, you can observe mitosis. Haploid cells, what are the examples? Male gametophyte is what? Pollen grain, right?
Pollen grain.
Yeah.
Pollen grain.
Take down. In both male gametophyte, yeah, male gametophyte is pollen grain. Female gametophyte is embryo sac. Okay. Female gametophyte. So, in male gametophyte, if you see, that is in pollen grain division happens, mitotic division happens, and this divides into two celled condition where it forms a vegetative cell and a generative cell. And generative cell again undergoes mitotic division, forming out two male gametes, and here will be vegetative cell. Understood? So, in pollen grain mitotic division happens, it forms generative cell and vegetative cell. Generative cell again undergoes mitosis, forming out two male gametes. Okay. And here if you see, for the formation of female gametophyte, that is embryo sac, haploid megaspore is there, right beta? Haploid megaspore undergoes 3 sequential mitotic divisions to form this embryo sac. Okay. Haploid megaspore undergoes how many mitotic divisions? 3 sequential mitotic divisions to form a haploid embryo sac. Fine. Embryo sac is how many celled condition? 7 celled? Yeah, 8 nucleated condition. As such, connected questions will be asked. So, this is a connected dot for sexual reproduction of flowering plants, which is in your NCERT 12. Okay. As such, you have to prepare. Now, we can move on to the karyokinesis and cytokinesis of mitosis. Meanwhile, take down.
Can you explain the honeybee case?
Hmm?
Can you explain the honeybee case?
Definitely. Meanwhile, you take down. So, in honeybee what happens is, the sex determination is not done by male or female, it is not showing any male heterogamety or female heterogamety. Based on the number of sets of chromosomes, based on the ploidy especially, gender is decided in the honeybees. Okay. So, those cells which are haploid, which are just having half the set of chromosomes, are considered as drones or males. Okay. Those honeybees which have complete set of chromosomes, they are called as what? Female honeybees. Okay. So, those female honeybees, after undergoing meiosis, they will again produce 2 haploid sets, right? Those are male honeybees. Again they will undergo mitosis and produce male honeybees as such. Okay. So, when we are dealing with the sex determination topic, I will explain you more clearly. As of now, till here it is enough now. Okay. Any other doubt here? Okay. Tell me if you are having any other doubt. Shall we move on to karyokinesis? Yeah. So, karyokinesis is having how many substages here? 4. Karyokinesis. Division of nucleus you can say, or movement of nucleus. Here we have how many substages ma? Prophase, metaphase, anaphase and telophase. Okay, 4 substages we have. First is what? Prophase. In prophase, what are the major events that are happening? Listen. First is chromosomal condensation. Okay, first change is chromosomal condensation. That is also called as chromosome untangling or detangling of chromosomes. Okay, and second is disappearance of nuclear envelope and nuclear membrane. Complete disintegration will not happen. Disappear means they will not go anywhere. They will just lose their structural stability. Okay, they will disintegrate their structure. Okay, so these are the 3 major events that happen in the prophase beta. First event is what? Take down parallelly. What is the first event? Chromosomal condensation. Chromosomal condensation, they will be forming some thread like structures called as what? Chromatids. Yeah, this chromatids will be adjoined to the nuclear membrane. They will be touching to the nuclear membrane. So this stage is also called as spireme stage. Spireme stage. Fine. And then nuclear membrane, yeah, nuclear envelope will disintegrate. Yeah, disintegrate or disappear. Fine. And then nuclear membrane and nuclear envelope will disintegrate. Most importantly, the centrosomes which are there, centrosomes which got duplicated in the S phase, which are present in the cell's cytoplasm, they will move to the opposite poles. Okay, centrosomes will move to the opposite poles. Now they are called as asters. Asters will be radiating out the spindle fibers. Fine. So centrosomes move to the opposite poles. Centrosomes moves towards the opposite poles, and here the asters will be radiating what? Spindle fibers.
In progress only they are attached to the spindle fiber.
Yes. In prophase, they are releasing out or radiating out the spindle fibers beta. In the metaphase, they will be attaching to the kinetochores. Okay. Asters radiate out spindle fibers. Fine. These are the 3 major events that happen in the prophase of mitosis. Clear, right? Now, amongst these phases, which phase is marking the start of metaphase? Okay. So now these are the events that are happening, and one thing you have to remember, before the start of karyokinesis only in our NCERT, he will clearly mention there are no clear cut lines to draw in between the stages. Okay, we can only see these are the events that are happening framed out under this stage, and there is a transition from this stage to this stage, right? So which stage, which event can mark the start or beginning of the metaphase? It is disappearance of nuclear envelope completely will mark the start of metaphase. Complete disappearance of nuclear envelope will mark the start of metaphase. So here you see ma, yeah. Complete disappearance of nuclear envelope will mark the start of metaphase. And in metaphase, chromosomes get short and thick. Chromosomes will become shortest and thickest, and they will align at the equatorial plate. That is, they will come at the center like this. Okay, that phenomena is called as congression. Okay, that phenomena is called as congression. Fine. All the chromosomes will come to one plane, that is equatorial plate. That phenomena is called as congression. And as we have discussed, to the each centromere, what are present? 2 disc shaped structure called as kinetochores are present. To this kinetochores, the spindle fibers will be attached. Okay. To kinetochores, what are attached? Spindle fibers are attached. Cores get attached to spindle fibers. Clear till here, right? Next is what? Anaphase.
Metaphase is best study to of chromosomes.
Yes. Metaphase? What did you say?
Studying of chromosome.
Observing chromosomes is better than this.
You can clearly see the structure of chromosome in the metaphase. Okay, structure of chromosome is clearly visible in the metaphase. You can clearly study the structure of chromosome. Yeah, and chromosomes are of what shape ma? Thickest and shortest. In prophase you can just see the thread like structures called as chromatids. They are thickest and shortest in, yeah, fine. In anaphase, anaphase is marked with what event? Splitting of centromere. Centromere splitting will happen. So now imagine like this, two asters are there at the opposite poles. What are these asters? Centrosomes. They will radiate out the spindle fibers. The spindle fibers got attached with what? Kinetochores. Now these spindle fibers will pull the chromatids towards the opposite end. Can you see two sister chromatids in the metaphase? Two sister chromatids are observed in the metaphase only. These sister chromatids will, centromere splitting will happen and they will be separated during the anaphase. As such, you can see different shapes of the chromosomes. What are the different shapes of the chromosomes? V shape, L shape, J shape and I shape. Okay. What are the different shapes? These are the shapes. V shaped chromosomes have two equal arms of the chromatids. They are called as what? Metacentric chromosomes. Fine. And L shape has one short arm and one long arm. These are what? Submetacentric chromosomes. Yes. And J shape, one very short arm and one very long arm. These are what, Nana? Acrocentric. Okay. And I shape, if you see, one very small negligible arm and one very long arm. These are what? Telocentric chromosomes. In anaphase. Okay. So shapes of the chromosomes are clearly visible in anaphase. Clear, right? Any doubt anywhere? No. So all three events of karyokinesis are clear to you. Next is telophase. So in telophase you see, what happens is, in the very first stage what happened ma? Beginning it is nuclear membrane and nuclear envelope got disintegrated. Here nuclear membrane and nuclear envelope are reformed. They reappear during the telophase. Nuclear membrane and nuclear envelope will reappear during the telophase. That is the first event.
Cytoplasm also divides.
Cytoplasm divides after telophase. That I will explain you. Okay. Have patience. So here nuclear membrane and nuclear envelope are formed, reappear during the telophase. Second is chromosomes get tangled again into chromatin. Okay. Here chromatin gets converted to chromatids. Here chromatid is converting back to what? Chromatin. Got it? So in telophase what are the major events ma? Telophase. Reappearance of, yes, reappearance of nuclear membrane and nuclear envelope. And this is the first event. And chromatids get tangled back. Chromatids tangled to chromatin. Fine. Chromatids get tangled back to the chromatin. Here it is detangling. Okay. Here it is tangling back. Once I will give you 2 minutes of time. Look into the all transitions properly. If you are having any doubt anywhere, you can ask me better. Revise it completely and ask me any doubt. Anjan, what you want exactly, beta? What you need exactly, you just ask me. I will repeat it again.
also the chromosomes are thickest only.
It will be the same, but shapes will alter. It depends on the length of the chromatid.
Difference between chromatid and chromatin.
Chromatid is these thread-like structures, beta. Chromatin is this. Just a second. This is chromatin.
Okay.
Thread-like structure is called chromatid.
Network is chromatin. Network of genetic material. Fine. Shall we move on to cytokinesis? Yes. See here. Cytokinesis is movement of cytoplasm, you can call, or simply you can call is separation of cell or formation of two daughter cells will happen. It is different both in animal cell and plant cell. First we will look into the animal cell. In animal cell, it is very simple. Cytokinesis is very simple in animal cell. What happens is, from the peripheral ends, a cleavage furrow is formed. This cleavage furrow is also called as cell furrow. Cell furrow or cleavage furrow will be formed, and this will divide the cytoplasm into two daughter cells. Okay, cell furrow or cleavage furrow is formed. This will divide the daughters. This will divide a single cell into two daughter cells. Okay, so here in animal cell, through what the cell division is happening? Through the formation of cell furrow or cleavage furrow. Fine, cell furrow or cleavage furrow. This is from periphery to the center. You tell me whether it is centripetal or centrifugal. Morning I told you already. From periphery to center. Centripetal. It is centripetal mode of cell division. See, from periphery to the center, so it is centripetal beta. Centrifugal means from center to periphery. Okay. So, as the division is from periphery to the center, it is centripetal. Clear, right? Next is in plant cell. In plant cell, it is not that simple as how it happens in an animal cell. So, see carefully. First, what happens is, in between two cells, at the center, phragmoplast will be formed. Okay, that phragmoplast will be synthesized from Golgi bodies only. Okay, that is rich in calcium pectate and magnesium pectate. This phragmoplast will move towards the peripheral ends, forming a cell plate. Okay, phragmoplast will move towards the peripheral ends, forming what? Cell plate. This cell plate will mature to a middle lamella. Okay, that forms the primary cell wall between two plant cells. Understood? So, first what is forming at the center? If these two cells are there, at the center first what is formed? Phragmoplast is formed. Phragmoplast. This phragmoplast is synthesized from Golgi bodies. Yeah, these Golgi bodies will synthesize phragmoplast, right? So, they will be they are rich in what? Calcium pectate and magnesium pectate. Calcium pectate and magnesium pectate. Once this phragmoplast is moved towards the peripheral ends, it is called as what? Cell plate. Phragmoplast moving towards the peripheral ends is said to be cell plate. It is centrifugal because it is from the center to the periphery, right? Cell plate will mature to what? Middle lamellae. Middle lamellae is the common wall between two plant cells. And you know the formation of wall layers, right? We have discussed in the plant cell wall. Inner to the middle lamella, primary cell wall is formed. Inner to the primary cell wall, secondary cell wall is formed. So, close to the cytoplasm, it will be secondary cell wall, primary cell wall and middle lamella. Clear, right? And what is the common wall between two? Middle lamella is the common wall between two plant cells. Okay? Clear, right? Any doubt anywhere till cytokinesis?
The cytoplasmic connections, plasmodiaphyte middle lamella.
Plasmodesmatal connections will be present, but they will traverse all the 3 layers of the cell walls. It will traverse the middle lamella, it will traverse the primary cell wall, secondary cell wall, and it will connect the cytoplasms of 2 plant cells.
So it will still be present.
It will be present. Okay. So till here it is clear, right? So what all we have discussed today? Cell cycle, all the sequential events that happen from the increase of cell size to the formation of 2 daughter cells. Second is we have clearly noted down the 3 different cell divisions: amitosis, mitosis and meiosis, and then 3 stages of the interphase. What are the 3 stages? G1 phase, S phase and G2 phase. We have marked out the events properly, and G0 phase also we have discussed, which is the quiescent center, and then karyokinesis and cytokinesis, all the events of mitosis we have discussed. Fine. And in our next class we will be clearly dealing with meiosis. Most importantly, in meiosis prophase 1 we will be highlighting. After that we will be looking into the numerical problems of applying both mitosis and meiosis, and then we will look into the significance of mitosis and meiosis. We can expect a statement based question from significance area. Okay. So this we will be looking in the next class. So that's all for today, and thank you. Any doubts anywhere you can approach me. Online students, you can drop a message in the chat box. I will be addressing your doubts. Okay. Thank you. So Abdul, you need metaphase. Abdul, listen carefully, beta. So here you see in metaphase what happens is, metaphase stage is marked by the beginning of what? Complete disappearance of nuclear envelope will mark the beginning of the phase, that is metaphase. Second major event is all the chromosomes which are formed, which has 2 sister chromatids, they will come at the center. Okay, at the center they will come. So this alignment of the chromosomes at the equatorial plate at the center is called as congregation. Okay. So 2 disc shaped structures which are present around the centromere, to this these disc shaped structures are called as kinetochores. To this, spindle fibers will be attached by the asters. Okay, and then splitting takes place, centromere splitting takes place in anaphase. So these are the events that happen in the metaphase, and most importantly the chromosome structure will be thickest and shortest. As such, you can clearly see the chromosome structure in metaphase. Okay, clear Abdul? Respond ma.
What is the difference between prophase, early prophase and late prophase?
Yeah, thank you beta.